Good evening, everyone. Thank you so much for being here tonight. So I would like to tell you a little bit about this project I've been working on for the last few years, which has kind of been like my baby. And it's been about teaching cancer cells how to die. But before getting to the core of the project, I think it's good to take a step back and see why cancer is so hard to fight. So, as you know, our bodies are made of cells. And if you take one of these cells and you look it up at the microscope, this is pretty much what it looks like. So cute, right? Maybe a little bit dumb, but ultimately pretty harmless, living peacefully with its neighboring cells. But life is also full of guilty pleasures, like smoking and drinking a bit too much, binge-watching TV series while eating deliciously unhealthy food. That's my favorite, at least. Exposing yourself to radiations or dangerous chemical poisons for fun. And, of course, the old god-fashioned, your DNA just getting damaged for no good reason. So one or a combination of these unhealthy habits can become a weapon that will ultimately transform your cute, healthy cells in dangerous cancer cells. But after all, what's the big deal if one little cell becomes a cancer cell? Well, the problem is cancer cells really, really like company. And they start to replicate, and then eventually they also start changing, and then they replicate, and then they change again, and then they start also invading the rest of the body. So they are actually a little bit like when you invite an Italian at a party, you know? You invite one, and then 3 of them comes, and then at the next party you have 9, then 27. By the 4th party, everybody is speaking Italian. I am not comparing Italians to cancer. I'm just saying we have a similar growth pattern. But doctors and researchers have been working really, really hard to cure cancer, and they come up with very, very innovative ways to make cancer therapies. But because cancer is so unpredictable in its way, in the way it changes, then some of these cells will be killed with the cancer therapy, but some are able to resist it. And that's a problem because these cancer cells that are able to escape the therapy, then they will eventually start growing back. And that's why some patients seem to be cured, and after a few months or a few years, they come back with a very aggressive tumor. So, of course, we want to find out what is the difference between these cells that are sensitive to the therapy and these cells that are resistant. And one of the first things that researchers looked into was the DNA of these cells. The DNA is the instruction manual that cells use to build themselves. So if there are typos there, for sure you will have different cell types. And yes, researchers did find some differences in the DNA, but they were not really enough to explain these differences. And if you think about it, our body, like cells in our body, already contains roughly the same DNA, but they look very different. For instance, we have muscle cells, neuron cells, blood cells, skin cells. So there must be another way that the DNA is regulated to decide which parts is used and which part is not. And that's the epigenome, one of my favorite things. Epigenome literally means on top of the genome. And you can imagine it as a series of doors that are either open or closed. And when they're open, it means that the cell uses that part of the DNA. And when they're closed, then that part is not used. It's completely discarded. And when we look at the epigenome of sensitive and resistant cancer cells, there are indeed differences. So the idea that my colleague and I had is, what if we can reprogram the epigenome of the resistant cancer cells and turn it into the one of the sensitive cancer cells? In this way, then the new sensitive cells could be killed with the initial therapy. And hence, we could basically teach them how to die. But how to do, to reprogram this epigenome? Well, that's quite a complicated thing. And that's also one of my favorite topic of conversation, so I could probably keep you here for the next 2 hours. But for the sake of time, I'll just give you the quick and dirty version of it so that we can all be here, out here quite soon. So you have your DNA, and you have the epigenome on top of it. And this epigenome determines which proteins the cells produce. And the proteins are the building blocks, the bricks that the cell uses to build itself. So a certain set of proteins will lead to a certain type of cells, for instance, resistant cancer cells. What if we had a drug that is able to change how this epigenome looks like? Then we would have different proteins and a different cell type, for instance, a sensitive cancer cell. So what we thought is, if we find such a drug, we could first give the reprogramming therapy to the resistant cells, turn them back to sensitive, and then the standard therapy would then be able to kill these newly sensitive cells. Well, that was a nice idea. Now the complicated part was to prove it. And bear with me for a moment. So to prove our idea, we took 4 different types of resistant cells coming from blood cancers. And then we combined them with 60 different types of reprogramming drugs, divided in 6 main mechanisms that were targeted. And we tried all possible combinations. So in total, I think it was 240 combinations, quite many. And we let each combination interact with each other for 9 days to give enough time for the reprogramming to happen. And after 9 days, because of course we cannot see inside the cell if it's a resistant or a cancer or a sensitive one, then we gave the standard therapy. And indeed, we observed that our idea did work. We were able to not fully sensitize all combinations, but there were certain ones that did work. And in particular, 2 types of mechanisms worked across all 4 cancer types. So this was pretty exciting for us because it was something new. And if you're wondering what is next after this, well, we have only proven this in the lab so far, and further studies still have to be done in order to bring this potentially to the clinic to help actual patients. But with this talk, I just wanted to show that reprogramming therapy does open new doors in cancer treatment. And, well, researchers are kind of like the explorers of the unknown, so this is definitely a new path worth exploring. Thank you.
Thanks a lot, Chiara. This looks like it took a lot of weekends and holidays in the lab.
Yes, it did, but I had company, luckily. So it was quite a big project. A lot of people collaborated. In particular, a colleague of mine, we started together, 2 bioinformaticians in a wet lab. I think many people thought it was a joke in the beginning. But yeah, in the end, we made it. And even in this presentation, I had help from a friend that made some of the images. And I think my boyfriend by now has an epigenetic drug overdose or something like that. But he's still alive.
Any questions from the audience?
Hi. Your estimation, when will we be able to defeat cancer? Thinking about it as a
That's a tough one. So I think there is a constant, when you hear people speaking about cancer, there's this constant gap of in 20 years we will, eradicate cancer completely. thing is, cancer changes. Like, you think you have one cancer and you have a bunch of different cancer cells, and the unpredictability of these cells is what makes it difficult to fight. But the good thing is that there are a lot of ways that people are trying to fight it, and this is just one of them. There are people trying to produce cancer vaccines or target it with nanoparticles. So I think that, I don't know the exact estimate, but a lot of people are working on this, so hopefully soon. 20 years. Just, you know.
Any more questions? Over there.
Has there been some unwanted symptoms of this treatment?
Yeah, so as I was saying, we only tested it in the lab, so we haven't actually tested it on humans. We tested it on human cells, but like in the lab. There might be some unwanted symptoms, but like with any kind of therapy, even chemotherapy has side effects. So that's why like testing needs to be like regulated, and it's not that, yay, we found a new drug, let's give it to a bunch of people. So there will be like certain steps. Some of these drugs have already been tested in humans, some have not. None of them, I think, has been tested in the reprogramming method. Like, they're more like given together with others. But
so maybe
yes.
we could get Huxer Pharmaceuticals to help us in testing
yes.
these.
All right. One more question. Yeah.
So just out of curiosity, these reprogramming drugs, did they have any side effects on the usual sensitive cells?
Sorry?
So basically, if you put reprogramming drug on a sensitive cell.
No, on the resistant one.
Yeah, but I mean, if you did.
Okay.
So would there be any effect on them?
There might be. That's
so you
why
didn't test that?
No, because, well, sensitive cells, it's difficult to have them because you've already killed them with the therapy. So the way that we get these cancer cells is because they are the ones that are left after the therapy. But of course, like, I think mouse or like animal experiments are a phase before going to human, and that's when we would know if the side effects on the sensitive ones are actually bad.
All right. Let's thank Chiara one more time.
Thank you.